there is no doubt in my mind that there are going to be several questions on the parts of the cell. And they do it in a couple different fashions. The majority of the questions, that model that's sitting on your desk will be at a station. There'll be tape on that model asking, either pointing to an organelle, and they ask one of a couple questions. They either ask you, name this organelle, or they ask you to name this organelle's function. The second type of question that they ask, it'll just be a card, an index card. And when you get to the index card, it'll just have a simple question on it, like it'll say, what organelle is considered the powerhouse of the cell? Right? They'll just give you the definition, and you've got to give me the organelle. So it would behoove us to make sure that we know all the different organelles that are found on this model so that we label them properly and then move along. Once this powers up, that's exactly what we'll do first. After that, we're going to pull out the microscopes. We're going to go through mitosis again, and we're going to start looking at mitosis under the microscope, because on the first lab exam, there will be questions where you have to look into a microscope, and you have to tell me what phase of mitosis that cell is in. So it's going to be important that you're able to do so. So we're going to prepare you for that event. All right, so let's reiterate a couple of things. Can I borrow your cell model, please? Yeah, no, it's one. So again, this cell model's coming back on the first lab exam. It's a guarantee. It's not a matter of if, it's just a matter of how many times. And how many times is really dependent upon the teacher that makes your exam. If he or she loves the cell, there'll be a lot of cell questions. If he or she kind of thinks the cell is kind of important, there'll be a handful of questions. It really does depend upon the teacher. What I do is I try to come in the day of the lab exam, and I try to more evenly distribute based on what I think is important and based on how I taught it. I'm going to spend a lot of time talking about tissues, which is the next thing that we're going to do in lab after today. So I'm going to try to make a lot of tissue questions on the lab exam. Having said that, we need to go over the parts of the cell. So remember that there's three major parts of the cell, those being the plasma membrane, those being the nucleus, and those being the cytoplasm. Every now and again that question gets asked. This is the basic model of a generic cell. So this picture and this model are pretty much the same thing. So what we're going to do is we're going to take this model step by step by step, show you each of the different organelles so that you can label them at your own tables. And then after this, what I've done is I've given you all the different organelles and shown you here's what each of those organelles do for a living. So but we're going to do all of these slides utilizing this picture. So first and foremost, if they put a sticker right here and asked you to name this part of the cell, what would you call it?
Cell membrane.
You'd call it the plasma membrane, right? Because that is the plasma membrane. As we look into the center of the cell, there is a roundish structure that is surrounded by white. It has two layers inside of it. This whole structure, from the white Swiss cheese looking structure inward, is collectively called the what? That is the nucleus. Having said that, nucleus, they'll never point to something and the question will be nucleus. What they'll ask is the part of the nucleus. So this white Swiss cheese portion of the nucleus is called the what? The nuclear membrane. Right? That is the nuclear membrane. Otherwise known as the nuclear envelope. What's the function of the nuclear envelope? We'll talk about this on Tuesday. Right, it has little holes called nuclear pores, which allows the movement of material in and out of the nucleus, right? As we go deep, the deepest portion of the nucleus, which is represented by this little orangey, reddish little ball in the center, that would be the what? That is the nucleolus. And what is the key function of the nucleolus? But you're getting closer. It makes ribosomes, and ribosomes make protein. So the nucleolus's job is to make ribosomes. So if they point to this organelle and they say, what is it? You would say nucleolus. If they point to this and say, what is this organelle's function? You would say it makes ribosomes. That's what it does. The last of the portions of our nucleus is the portion that's not the nucleolus and not the nuclear envelope. It's kind of a clear area and it looks like it has little blue springs in it. This is the chromatin. If they point to this, I will take any of the following answers. I will take chromatin and I will take chromosomes. The job of the chromatin and the chromosomes, this is where our genetic material is. This is where we replicate our DNA. This is where we produce mRNA. So we have the nuclear envelope, this white Swiss cheese looking structure. We have the chromatin or chromosomes, which is the next layer deep. And the deepest layer of all, this is the nucleolus. The nucleolus is where we produce our ribosomes. Everybody kosher with that. We also have, and I'm showing you two varieties of the same organelle. Right? This orange and brown organelle. This is the mitochondria. The mitochondria's function is to do what? It produces ATP. That's what it does. If you put powerhouse of the cell, I'm going to assume, especially on the first test, I will assume that what you meant was it's the powerhouse of the cell because the powerhouse of the cell produces ATP. I'm going to assume you know that that means it produces ATP, so you can put that. But at the end of the day, the mitochondria produces ATP. That's its function. This is showing the mitochondria cut in half, so you can actually see the interior of the mitochondria. This is showing the mitochondria in 3D, so what you're seeing is the outer covering. The key here, is anybody here colorblind? Because I've had colorblind folks in the past, and that makes this a little more difficult. This is one of the times where match the color, right? The colors do matter. So orange and brown, we're talking about mitochondria. Everybody kosher with mitochondria, everybody kosher with the nucleus and its 3 parts. Good. Anywhere where you see a white dot, each of these white dots represents a what? That is a ribosome. Every white dot is an individual ribosome. And the function of ribosomes is to do what? It makes proteins, right? So the nucleolus makes the ribosomes are what produce the protein. So every white dot represents a ribosome. Now, this lightish, bluish, kind of a light blue or a blue-green structure, we see one here, and then we see its dark blue interior over here. These are your endoplasmic reticulums. Now we have 2 varieties of endoplasmic reticulum. One of them was rough, and one of them was smooth, right? From Tuesday? Anywhere where you see endoplasmic reticulum that has white dots surrounding it, that's going to be the what endoplasmic reticulum? That will be your rough endoplasmic reticulum. On the lab exam, if they point to the rough ER, and you want to put rough ER, I'm all for it. If you want to see what my answer sheet's going to say, it will literally say RER, for rough endoplasmic reticulum. And if you want to put RER, I'm going to assume you know that RER means rough endoplasmic reticulum. On the contrary, if they point to the smooth endoplasmic reticulum and you want to put SER, that's what my answer sheet's going to say too. Or smooth ER, either way you're golden. So anywhere where you see endoplasmic reticulum, and it has these white dots, we're dealing with the rough ER. What is the function of the rough ER? It alters the proteins made by the ribosomes. Now here's the thing, if you put produces protein, I have to take it. I will be angry as I do so, but I've got to take it, because your book does literally say it synthesizes protein, right? So, and I have to go with what the book says. Although that's not true. It doesn't produce it, it just alters it and packages it, right? But that's the rough ER. Any place where you see endoplasmic reticulum, such as right here, where there's no white dots surrounding it, this is going to be smooth ER, right? So here's some smooth ER, and oh, here's some smooth ER. So we've got a couple different areas where there's endoplasmic reticulum that does not have any ribosomes. These are going to be examples of smooth ER. Everybody with me? And what's the function of the smooth ER? Produces a few things. Does a few things for the cell. It makes fatty acids. What else did y'all say? Detoxes the cell, right? It definitely helps it detox. It makes fatty acids, makes steroids. So it does a few different things. Pick one. Right? You don't have to give me a book. Just give me one thing that the smooth ER does, and we're in like one. Right? I'll stack a few of them in there to make sure that I cover all my bases. It detoxifies the cell. It produces fatty acids and steroids and cholesterol. And those are the basic functions we talked about on Tuesday when we talked about the SEO. The other organ, another organ that we talked about was the Golgi complex. That's this one right here. So anything that you see that is pink and whatever this whitish color are, we're going to go with tan. Right? This pink and tan organelle and the pink dots that we see running along the side of it, this is all representative of the Golgi complex or the Golgi apparatus. The key word I'm looking for there is Golgi. If you just put Golgi, we're good, right? What's the function of the Golgi complex? It shifts things around the cell. If you put train tracks in the cell, I'm good. All right? It ultimately takes proteins specifically, but other substances too. It takes the materials made by the cell and it transports them around the cell. So transportation, moving things around the cell, shipping proteins, all those basically are saying the exact same thing. That's the Golgi. I'm actually kosher with that analogy. And if it gets it right on the test, all the better. We then had another structure. We see a few of them. Here's two of them. They're purple in the center, kind of silver on the outside. Here's a third one in the cell. We're just seeing its outer covering. So purple and silver, that represents a lysosome. And what is the function of the lysosome? I was telling you that the lysosome is to the cell, but what organ is to our body? It's the stomach of the cell, because lysosomes have digestive enzymes. And so the cell uses lysosomes when it brings material into the cell, endocytosis, whether we're talking about phagocytosis or pinocytosis. Anything the cell brings in, it will merge it with a lysosome. Lysosome will dump its digestive enzymes, dissolving those solutes into smaller particles, just like our stomach takes food that we bring in and breaks it down into smaller components. So here we are talking about lysosomes. Everybody with me? Which brings us to this yellow and orange structure. Here's one in the center. Here's one showing the outer covering. These are both the same organelle. These are proteasomes. What do we talk about the proteasome doing? We talk about the proteasome helping to detoxify the cell specifically of free radicals. So the proteasome helps to break down free radicals within the cell. A free radical is a byproduct of cellular metabolism that are ultimately harmful to the cell. They're needing to be stabilized. The way they usually stabilize themselves is by attacking the nuclear, the plasma membrane, therefore harming and potentially destroying the cell. It's kind of like if you go to a tire factory, right? The tire factory is making some great things, making tires. But in order to do it, it's got a smokestack kicking out toxic stuff into our atmosphere, which is harmful, right? The proteasome's job is to take that smoke from the smokestack and not make it harmful to the cell. Does that make sense? Amen. Amen. Proteosome, proteasome.
Is there another name for that on the picture? Because I can't see it.
I think it's up there. Wait, is it proteasome? Up there, it's like a little purple thing right by the cilia, or over there. Yeah.
Right there. It's pointing to this. I'm telling you on this model it's this. Which leaves us with one more organelle. And that is this one right here. We just talked about these. These are the centrioles. So this is a centriole. And if they ask you what does the centriole do, you're going to say what? Aids in mitosis. In fact, if you just put the word mitosis, I'm going to assume that you understood that that's an organelle that helps in mitosis. So first things first. Take the model, as we've just shown you all the organelles. Make sure you label each of the organelles that we've talked about, Gesundheit. Take your pictures so that because again, it's great to have a picture in the lab book, because the lab book does a great job of giving you another look at the same thing. But that picture in the lab book will not be on your test. This, I promise you, is going to be on the test. You will come to a station and you will be looking at this on your first lab exam, and there'll be questions based on what we just went over, right? Both name that organelle and what does that organelle do for a living. So label this, take pictures so you can study it at home, and then we'll move on to mitosis afterwards. With regards to the things that all of these organelles do, that's what all of these slides are for. All right, as we get to centrosomes, centrioles, cilia, flagella, ribosomes, all the endoplasmic reticulums, it's all in the lab notes as well, giving you information as to what all of these organelles do. And if you need help, if you need me to, as I'm walking around, if you want to make sure that everything's labeled properly, of course, ask, and I'll be happy to help.